Welcome to part 2 of my 9th short talk on epigenetic science. In this 2-part series, we're exploring the fascinating way our cells access the information in the genes in our DNA to make proteins. In part 1, we discussed how the genetic code is read and the process of making a copy of a gene, which is called transcription, and it occurs in the nucleus of the cell. After a gene is transcribed into an mRNA copy, the mRNA has to be edited in the nucleus. One of the amazing editing processes is called RNA splicing. I'm going to discuss RNA splicing in another talk because it's extremely important for our epigenetics. Today, we're going to explore how the mRNA is used to make a protein in a process called translation, and this occurs in the cytoplasm of the cell. My main purpose in showing us the incredible processes our cells use to express our genes into proteins is so that when we think about applying our mind to epigenetics, we have a tangible reality about how cells use the genes to make proteins. Our lifestyle choices and our thoughts and emotions really do affect how our cells express our genes. My name is Robert Needleman. I'm a molecular biologist and biochemist. Welcome to part 2 of this episode of Empower My DNA. Let's go back to the mRNA copy of a gene. After the mRNA is edited in the nucleus, it leaves through a pore in the nuclear membrane. In order for the mRNA to be read into a protein, it has to be approached by a ribosome, the molecule shown here in brown. A ribosome is also known as a cellular protein factory. Ribosomes have a small subunit and a large subunit. Ribosomes are made of ribosomal RNA and ribosomal proteins. In order for the genetic code to be read, our cells use another RNA molecule called transfer RNA. Here is a transfer RNA. All of the 20 main amino acids are carried by tRNA molecules to the ribosome. Transfer RNA molecules are short RNAs. Every tRNA has an anticodon which can pair with the codon that codes for the amino acid it carries. The match between the amino acid and the anticodon is critical for the accuracy of the protein being made. Now we see the tRNA entering the ribosome. The messenger RNA is surrounded by the ribosome. Here's the codon CAC. A tRNA with the anticodon GUG has paired with it and will deliver its amino acid histidine into the growing protein chain. The next amino acid in the chain has the codon AGU, and the tRNA with the anticodon UCA pairs with it and brings serine, the amino acid serine, into the growing protein chain. This incredible process continues until a stop codon is reached and the protein is completed. Each amino acid being incorporated into the growing protein is based on the genetic code carried in the messenger RNA. The growing protein emerges from the ribosome and it begins folding. When the protein's completed, it's released from the ribosome and it continues folding into its more complex shape. The way our cells make proteins from our genes is truly wondrous. I hope you're as fascinated by this process as I am. Before we go, I have another excellent real-time animation from the DNA Learning Center. We start with the transcribed and edited mRNA inside the nucleus. The completed copy of the gene leaves the nucleus through a nuclear pore and enters the cytoplasm. There can be several million ribosomes in a cell. This is called a free ribosome in the cytoplasm. The blue molecule is the small subunit of the ribosome binding to the messenger RNA. Now, the purple large subunit joins and the ribosome is functional. Here we see multiple transfer RNAs arriving at the ribosome to deliver amino acids in the order dictated by the genetic code codons in the messenger RNA. The newly synthesized protein is shown here in red, emerging from the ribosome. Here are many transfer RNAs carrying all of the amino acids. A closer view shows different amino acids bound to tRNAs based on the anticodon. The match is critical, so the correct amino acid is added. The mRNA is pulled through the ribosome. Looking inside the ribosome, we can see the awesome system of tRNAs in 3 positions in the ribosome. This tRNA is arriving. This tRNA is delivering its amino acid, and this tRNA is exiting the ribosome. It's exciting to be watching a ribosome add about 2 amino acids per second. The mRNA is pulled through the ribosome, codon by codon. As more of the new protein emerges from the ribosome, it begins folding in a process called co-translational folding. When the protein is completed, it leaves the ribosome. Wow, I truly hope you find how our cells make proteins fascinating. This is going on right now in many of the cells in our body. Lifestyle choices we make have a big effect on our protein synthesis efficiency and on the kinds of proteins we make, which in turn affects our health. We can support ourselves by saying, I make positive lifestyle choices for my health and empowerment. Thank you so much for joining me in this 2-part series. The human body is truly miraculous, so it's worth our time and our effort to feed our body healthy foods and positive thoughts and emotions to optimize our health span. I hope to see you on my next talk, where we're going to discuss the Human Genome Project. It's a fascinating topic. Take good care and have a wonderful, healthy, empowered day.